This recording is the nervous system and nervous tissue, part 2 of 2. We know that there are different concentrations of ions in the extracellular fluid outside the plasma membrane of the cell and in the intracellular fluid in the cytosol of the cell. Because ions are charged atoms, these ions create a voltage due to the difference in the concentration of them. In understanding electricity, we need to understand a little bit about Ohm's law. Ohm's law tells us that current equals voltage divided by resistance, which simply means that the greater the voltage, the greater the current. The greater the resistance, the less the current. The voltage in a plasma membrane can actually be measured, and resistance is provided by the plasma membrane. All living cells maintain this difference in concentrations of ions across their membranes. When we're measuring the voltage, it's literally the difference in the concentration of ions just external to the plasma membrane versus the concentration of ions directly internal to the plasma membrane. We know that there are different concentrations of ions, which are charged atoms, outside the plasma membrane versus what's inside the plasma membrane in the cytosol. There tends to be a higher concentration of sodium in the extracellular fluid outside the cell and a higher concentration of potassium in the cytosol. These ions normally can't pass through the plasma membrane. They need help. And help can come in the form of protein channels, which are selective in terms of which ions they will allow to pass from one side of the membrane to the other. These proteins embedded in the plasma membrane acting as channels include chemically ligated or ligand-gated channels, which open when the right chemical binds to them, changing their shape, allowing ions to cross the membrane, thereby changing the charge. These channels can allow ions to enter or leave the cell. Mechanically gated ions will open when there's some kind of physical deformation, such as pressure, that can change the shape of the protein that will allow these channels to open or close, again allowing ions to enter or leave the cell. Here we see an example of this on the lower right-hand side of the slide. Some protein channels are voltage gated, which means they can open and or close in response to changes in membrane potential. Normally, the inner portion of the plasma membrane has a negative charge. Leakage channels are protein channels that can open and close randomly, contributing to the resting membrane voltage of excitable membranes. The membrane potential is due to the distribution of charges across the cell membrane. This can be measured in millivolts, as you can see in the picture at the top on the left. It compares the charges inside to the charges outside. With a net negative charge of negative 70 millivolts being the normal measurement for neurons. Other cells can have other membrane potentials. Negative 70 millivolts is the resting membrane potential for neurons. That means at rest, a neuron will measure at negative 70 millivolts. Because the membrane surface has this slight difference in ion charges, this allows neurons and muscle cells or muscle fibers to generate electrical signals. We've already seen this with skeletal muscle fibers when they respond to an incoming nerve impulse from a motor neuron. Resting membrane potential is the description of a membrane at rest. It's not being stimulated to do anything. The membrane is already in a state of being polarized. The membrane is polarized because leaky channels that we saw in one of the previous slides allows potassium to leak out, and in smaller amounts allows sodium to leak in. This allows the generation of more negativity just inside the plasma membrane and more positivity just outside the plasma membrane, resulting in a potential difference that's measured at negative 70 millivolts in a neuron. The resting membrane potential, even though it's established by potassium leaking out, is going to be maintained by sodium-potassium pumps. Remember, there are different ions involved in the extracellular fluid as well as the intracellular fluid, which is the fluid portion of the cytosol. If you look at this picture on the left, you'll see that sodium exists in a higher concentration externally. Potassium exists in a higher concentration internally. However, there are some sodium ions internally and some potassium ions externally. There are also chloride ions externally and phosphate groups and protein molecules inside the cell. When everything is added up with its positive and negative charges, again, the inside of the cell tends to be more negative and is measured at rest in a neuron at negative 70 millivolts. The sodium-potassium pump maintains a higher concentration of sodium on the outside of the cell and a higher concentration of potassium on the inside of the cell membrane because it pumps out 3 sodium ions against their concentration gradient while pumping in 2 potassium ions, also against their concentration gradient. This requires energy in the form of adenosine triphosphate. And remember that the cells are polarized. Polarity occurs because there is a net difference in electrical charges with the external portion of the plasma membrane being more positive and the internal portion where the measurement is taken being measured as more negative. If there were equal amounts of charges on both sides of the plasma membrane, there would be no excitability and there would be no polarity. The polarity only exists because there's a difference between positives and negatives. And you remember that opposites attract. We know that action potentials are electrical impulses. In neurons, we call them nerve impulses. We know that the membrane of a neuron at rest can be measured at negative 70 millivolts because it is polarized. If a stimulus is applied, that stimulus can potentially open up protein channels that will allow the influx of sodium. Because sodium has a positive charge, that makes the inside of the membrane of the neuron less negative, depolarizing it. And if depolarization reaches negative 55 millivolts, At that point, the membrane has reached threshold. When threshold value has been reached on the membrane, that change in the voltage initiates an action potential where voltage-gated sodium channels will open up, allowing a very rapid influx of sodium into the cell just inside the plasma membrane in the cytosol. Sodium will depolarize the membrane until it reaches positive 30 millivolts. At positive 30 millivolts, technically sodium voltage-gated channels become inactive. However, since no sodium is passing when they're inactivated, we're simply going to say that sodium voltage-gated channels close at positive 30 millivolts. There's something else that happens at positive 30 millivolts. At positive 30 millivolts, potassium voltage-gated channels will open. Potassium is also positively charged. And when potassium voltage-gated channels open up, potassium will move out of the cell, taking its positive self with it, repolarizing the membrane, because as positive potassium exits, the membrane becomes more negative internally. Because potassium voltage-gated channels close more slowly than the sodium voltage-gated channels, too much potassium exits, and the cell membrane at that area becomes hyperpolarized. Here we are at resting membrane potential, and now we've gone past that. Basically, in order for the membrane to reestablish itself to negative 70 millivolts, the sodium-potassium pump will take over that job. Here's another illustration of an action potential. I'm going to disappear from your screen so you can read the slide a little bit better. Action potentials are very rapid events. They only take 2 milliseconds to occur. Depolarization starts with the opening of sodium channels with stimulus, allowing sodium to come in. However, once threshold is reached, sodium voltage-gated channels open and sodium rushes into the cell, changing the membrane potential, making it less negative. When the membrane potential reaches positive 30 millivolts, sodium voltage-gated channels close and potassium voltage-gated channels open. This allows potassium to exit the cell, and when potassium exits the cell, that means that it takes its positive self with it, making the inner portion of the membrane more negative, going beyond negative 70 millivolts and hyperpolarizing because it's gone beyond resting membrane potential. When action potentials occur, they occur as an all or none principle. What this basically means is that a stimulus will either be strong enough to stimulate an action potential because it's reached threshold, or it won't. Once threshold is reached, an action potential will occur. It is an all or none principle. It's kind of like flushing a toilet. Once a toilet is flushed, It can't get the water back. It's also the same thing as a cannon firing. Once that cannon fires, there's no way to get the cannonball back as it's traveling through the air. The magnitude, which is basically the amplitude of an action potential, is always going to be the same. Understand that the sodium influx required to reach threshold only produces about a 0.012% change in the intracellular sodium concentration. That's how small of a change needs to be produced to reach threshold. And these small ionic changes are very easily corrected because an axon membrane has thousands of sodium-potassium pumps. Once an action potential has been generated, there is a period of time where a second action potential cannot be initiated or generated. This occurs during depolarization and part of repolarization. This period of time, which you see in green on this slide, is known as the absolute refractory period, because there is absolutely no way that a second action potential can be generated, no matter how strong the stimulus. However, after a certain point during repolarization has been reached, depending on the strength or relative to the strength of the stimulus, there may be a second action potential or subsequent action potential generated during this period of time called the relative refractory period. The reason this happens is because it takes a while for the sodium voltage-gated channels to reset themselves enough in order for that second action potential to occur. So the relative refractory period is relative to the strength of the stimulus, and the membrane will respond only to a very strong stimulus during the relative refractory period. All action potentials are alike. Once they start, they go, and they have a very specific amplitude. In other words, the stronger the stimulus does not mean that there's going to be further depolarization beyond positive 30 millivolts. The amplitude or height of action potentials is always the same. If we look at the picture below, you'll notice that there has been stimulus applied, but the stimulus does not reach threshold. Therefore, there is no action potential. In the second example, where there has been stimulus applied, because it is a small stimulus, the frequency of action potentials is noted here. Look what happens when the stimulus is stronger. You'll notice a stronger stimulus on the third example and more frequent action potentials. That's how our nervous system codes for the intensity of the stimulus. Stronger stimuli will generate more frequent action potentials. So it's the frequency that lends to the perception of the strength of a stimulus, not the amplitude of the individual impulses or action potentials. In order for an action potential to occur, there has to be reception of a stimulus. We know that reception can occur on the membranes of dendrites as well as on the membranes of soma, which are the cell bodies of neurons. Most commonly, it's the dendrites. It can occur on the soma. However, the area where an action potential is generated is going to be the axon hillock. That's the initial segment of the axon. The axon hillock and axons contain a high density of voltage-gated sodium channels, and therefore they can depolarize very rapidly. As depolarization spreads, more sodium voltage-gated channels open, and the spread of this electrical impulse will travel from the axon hillock down to the axon terminals. The direction never moves backwards. It always goes from the axon hillock towards the axon terminals in one direction. Speed can increase in the spread of this impulse according to the diameter of the axon. The larger the diameter, the faster the speed. The smaller the diameter, the slower the speed. If temperature increases, that can increase the speed of the propagation or spread of this electrical impulse or nerve impulse or action potential down the length of the axon. If myelin exists, that can also increase the speed at which the action potential will be carried down the length of the axon. Remember, some axons are very long. The axons for motor neurons that go from your spinal cord down to your toes can be anywhere from 3 to 4 feet in length. When we look at what happens in terms of spreading this electrical impulse on an axon that has no myelin, unmyelinated axons undergo a process of conducting this electrical impulse that's called continuous conduction. It's called continuous conduction simply because as positive charges move inward, there's depolarization that continues on every segment of the axolemma, which is the membrane of the axon. This continuous spread of an action potential goes uninterrupted. When myelin is involved, it can go much faster because every part of the membrane does not have to depolarize. There is an excellent video that I've put in Blackboard for you to watch. You'll find it under the Lessons tab. If you click on Lessons and go all the way down past all the units, look for a folder that says Additional Resources. When you click on that folder, you will see more videos, and there are lots of great videos showing me explaining different pictures, as well as this particular video, which is how action potentials are propagated. It is an animated video, and I recommend that you watch it. It's excellent. Again, action potentials with continuous conduction can occur faster when the diameter of the axon is larger and when the temperature is higher. With myelinated axons, we have faster propagation, but that is not continuous conduction. Instead, propagation of an electrical impulse or nerve impulse in myelinated axons is referred to as saltatory conduction. The Spanish verb saltar means to leap or to jump. With myelinated axons in the peripheral nervous system, where we have these little nodes of Ranvier or myelin sheath gaps, the only portion of the axon that is required to undergo depolarization is here at the node of Ranvier. From there, the electrical current can simply flow to the next node of Ranvier, where depolarization will occur. Again, the electrical change will flow and depolarization will occur. This means that the conduction with saltatory conduction in myelinated axons is much faster. In fact, it's up to 30 times faster. Because it appears to leap from node to node, it's been given this name of saltatory conduction, and this actually helps to save energy in the form of adenosine triphosphate because less surface area is going to be required to reestablish the membrane potential by the sodium-potassium pumps. We know that in order for an action potential to occur, the voltage on the membrane of a neuron has to be taken from negative 70 millivolts to its threshold value at negative 55 millivolts. When local changes in membrane potential occur due to small stimuli, that will cause a small depolarization in that locality on the cell membrane that over time will simply disappear due to the sodium-potassium pumps reestablishing resting membrane potential. This stimulus simply wasn't strong enough to take the membrane to threshold. Here we have a larger stimulus that causes more depolarization, yet is still not large enough to hit threshold. In this particular circumstance, we've got a longer lasting stimulus that's still not enough to depolarize the membrane to negative 55 millivolts. And then finally, here we have stimulus that is at or above threshold, triggering an action potential. So graded potentials are merely local changes in membrane potential that usually occur in dendrites, can occur on the soma. The amount of change in the voltage is going to be determined by the size of the stimulus. One of the things you need to know about graded potentials is they can either be depolarizing or hyperpolarizing. If they're depolarizing, they're taking the membrane closer to threshold. If they're hyperpolarizing the membrane, they're taking it farther away from threshold, which makes it less likely for an action potential to occur. If sodium or calcium enter, crossing the plasma membrane into the cytosol adjacent to the membrane, that will depolarize the membrane. If potassium leaves, making the inside more negative, or if chloride ions enter the cell, again making it more negative, that hyperpolarizes the membrane. If it's a small event, it dies out because the sodium-potassium pumps reestablish the membrane potential. The larger the hyperpolarization in a graded potential, the more the sodium-potassium pumps have to work to reestablish resting membrane potential, and the stronger the stimulus would have to be to change the direction of the membrane when it is polarized to this extent, we'd need a much stronger stimulus to get it to the point of threshold. If we look at the chart below on the right, you can see the comparison of graded versus action potentials. Graded potentials are a graded response depending on the size of the stimulus. Action potentials have an all or none response. They're either going to go, and when they go completely, or not. Graded potentials decrease with distance. Action potentials propagate over the entire cell. Graded potentials have no refractory period. Action potentials have a refractory period. Graded potentials can be added up in a process known as summation. Action potentials cannot be added together. There is no summation. Graded potentials are triggered by stimulus, for example, a ligand or mechanical deformation. Action potentials are triggered by the membrane reaching its threshold value. There are two types of graded potentials. Generator potentials are generated directly on the receptive regions of sensory neurons, including those that have both free nerve endings and those that have encapsulated nerve endings. So generator potentials are generated directly on neurons. However, receptor potentials occur when special receptor cells are involved. This is seen in Merkel cells or tactile cells that are found in the stratum basale. When those cells are stimulated, they will experience graded potentials on their membranes, resulting in the release of neurotransmitters that will communicate with sensory neurons. We see receptor potentials occur with cells not only in the skin, in the epidermis, but also those cells involved in taste, as well as photoreceptors found in the retina of the eye. Receptor potentials only happen in cells that act as receptors, which in turn communicate with sensory neurons. On this slide, we've got a really nice illustration of the difference between a generator potential, which occurs directly on a neuron, where the stimulus causes depolarization of that membrane, and if it reaches threshold, that will generate an action potential, which is a nerve impulse. On the right-hand side, we see a great example of a receptor potential because we have a special receptor cell that is going to receive the stimulus. When the stimulus affects the membrane of the special receptor cell, that will cause the voltage on the membrane to change, allowing calcium to enter the cell, triggering the cell to release its vesicles containing neurotransmitters into a synaptic cleft between the receptor cell and the neuron. It's a more indirect method of communication. So generator potentials are generated directly on sensory neurons. Receptor potentials are generated on specialized receptor cells, which will then communicate with sensory neurons. Our nervous system works to have communication between neurons. It's not just one neuron involved in a length of communication. Communication often involves at least more than one neuron, at minimal 2 neurons. So if we look at a chain of neuronal communication where we have one neuron communicating with a second, we call the first neuron prior to the synapse with the second the presynaptic neuron. The second neuron, which would be positioned after the synapse to receive the neurotransmitters released by the first neuron, would be referred to as the postsynaptic neuron because it is positioned after the synapse. And this area would be representative of the synapse between the two, where neurotransmitter will be released. Now, when the first neuron, the presynaptic neuron, releases its neurotransmitters, that will cause graded potentials in the dendrites, which I didn't draw in my picture, of the postsynaptic neuron. Those graded potentials may bring the postsynaptic membrane towards threshold, in which case they would be known as depolarizing, or it can take them away from threshold, which would make those graded potentials hyperpolarizing. Let's take a closer look at this picture to the left. On the left, you see a large postsynaptic neuron. If you look closely, you'll notice the synaptic end bulbs or axon terminals marked in green and red, communicating with the dendrites and the soma of the postsynaptic neuron. They're colored green and red for a reason. If we look at the chart underneath the picture, you'll notice that the signals coming from the green synaptic end knobs are taking the postsynaptic membrane towards threshold, from resting membrane potential at -70 to -55 millivolts, whereas the red synaptic end bulbs are taking the postsynaptic membrane away from threshold. And the postsynaptic membrane cannot fire unless it reaches threshold. Because some of these signals take the membrane towards threshold through the process of depolarization, we refer to those as excitatory postsynaptic potentials, because they're potentially going to cause the cell to move toward threshold where an action potential will occur, which will allow a nerve impulse to occur in the postsynaptic neuron. So excitatory postsynaptic potentials move the postsynaptic membrane towards threshold. Inhibitory postsynaptic potentials are identified by the red synaptic end bulb. Those synaptic end bulbs are releasing neurotransmitters that are taking the membrane away from threshold and are therefore inhibiting the possibility of reaching threshold and a resultant action potential occurring. So inhibitory postsynaptic potentials cause the membrane of the neuron to move away from threshold. Now, the cool thing about these excitatory and inhibitory postsynaptic potentials is that they can all be added together, and they're added together at the axon hillock. Basically, it's like an arm wrestling match. The strongest signals are going to win out. So if there's enough excitation to bring the postsynaptic membrane to threshold, then that's what wins. If there's enough inhibition to take it away from threshold, then that's what wins. On the previous slide, we saw a picture of a postsynaptic neuron receiving information from other presynaptic neurons. And when there are different areas of a neuron being stimulated, those signals can be added together, which is the process of summation. All of these signals are going to be added together at the axon hillock, except for in sensory neurons. This is where the combined effects of these different graded potentials, whether they are hyperpolarizing or depolarizing, is going to be added together. If the membrane depolarizes to the point where it reaches threshold, we know an action potential will be initiated. Summation can either occur spatially in different places, like we saw in the previous slide, or in one place on a dendrite or soma where signals are very quickly generated over a short period of time. That is referred to as temporal summation. Let's quickly review the events at a synapse. Understand that a synapse is where a neuron and another neuron, or an effector, which is a muscle or a gland, will communicate. It's not a physical connection because at the synapse there is a space between these structures known as the synaptic cleft. When an action potential, which is an electrical impulse or nerve impulse, travels down the length of an axon, the change in voltage will cause calcium voltage-gated protein channels to open up, allowing calcium to enter into the synaptic end bulb. This influx of calcium triggers the synaptic vesicles in the synaptic end knob to move towards the membrane of the synaptic end bulb in order for their membranes to fuse, releasing the neurotransmitter into the synaptic cleft by way of exocytosis. The neurotransmitter will then diffuse across the synaptic cleft to bind to ligand-gated protein channels. Once the neurotransmitter binds to the channel, it will cause the channel to open up. In this case, we see sodium moving in just inside the postsynaptic membrane. That would cause depolarization, and if enough depolarization occurs, an action potential will be generated by the postsynaptic neuron. There are different types of synapses. With cells that are joined together by gap junctions, that allows for electrical currents to flow between the cells through the gap junctions, and this forms an electrical synapse that's commonly found in cardiac muscle as well as single unit or visceral smooth muscle. Remember, in cardiac muscle, not only do we have gap junctions to allow the electricity to flow, we also have desmosomes. The gap junctions and desmosomes in cardiac muscle form structures known as intercalated discs. This picture is actually a picture of single unit or visceral smooth muscle, where we have gap junctions and tight junctions. Allowing an electrical current to flow directly from one cell to another allows the cells to function in sync with one another. With the electrical flow in visceral smooth muscle, that allows for a contraction to occur that is referred to as peristalsis. It's a wave-like motion. In cardiac muscle, we know that the chambers of the heart beat as a functional syncytium. We've covered chemical synapses, and here's another example of that. With chemical synapses, it involves the transmission of a chemical from one cell to another. The release of acetylcholine into the synaptic cleft is an example of one chemical synapse. With neurotransmitter systems, the effect of the neurotransmitter is dependent entirely on the receptor. I'll say that again because this is really important. The effect of the neurotransmitter is not due to the neurotransmitter. It's dependent on the receptor and how the receptor responds to the neurotransmitter. There are two classes of receptors. Ionotropic receptors are generally ligand-gated and tend to allow the flow of ions through the proteins when the receptors are bound with their appropriate molecules. Metabotropic receptors involve a much more complex system of proteins that, when activated, result in metabolic changes inside the cell. Metabotropic receptors are involved in a system called a second messenger system. We'll take a look at that. Metabotropic receptors are involved in second messenger systems. Second messenger systems have the effect of causing a lot of chemical reactions within the cell. We know that metabolism is the sum of all chemical reactions in the human body, hence the term metabotropic, meaning that there's going to be a lot of chemical reactions as a cause or as an effect of a molecule binding to a receptor. Because what happens in a second messenger system is a signal molecule acts as a first messenger, which can bind to a G protein linked receptor. This purple molecule is the G protein linked receptor. Once these two bind together, that activates the G protein, which actually travels along the membrane to activate an enzyme. Once the enzyme is activated, it produces a second messenger molecule that will further act to activate another enzyme, which will cause chemical reactions within the cell. There are two common messenger systems, second messenger systems that use cyclic adenosine monophosphate and inositol triphosphate. You really do need to know the names of these second messenger molecules: cyclic adenosine monophosphate and inositol triphosphate. As I mentioned earlier, the effect that occurs at a synapse is really dependent upon the receptors rather than on the neurotransmitter that's released. Nicotinic receptors tend to cause depolarization, whereas muscarinic receptors can either cause depolarization or hyperpolarization. I really like this picture because it's a great way to illustrate how the receptors on different organs will have different results, even though a particular neurotransmitter is released. Let's look quickly at the autonomic nervous system. If you've heard of the sympathetic division of the autonomic nervous system, you may have heard the term fight or flight. If a person encounters a bear and has to run away from the bear, we see that there are two neurons involved in the sympathetic division. The presynaptic neuron releases acetylcholine. However, the postsynaptic neuron releases norepinephrine. This norepinephrine that is released will go to different organs. When the sympathetic nervous system is stimulated, the receptors on the heart are going to respond to norepinephrine in a manner that causes the heart to contract more rapidly and with more force. Whereas the receptors in the salivary glands are going to cause the salivary glands to decrease their production of saliva. That's why if a person is nervous and has to get up in front of an audience and speak, he may experience cotton mouth because his salivary glands start to slow down a great deal. If you think about it, if a person encounters a bear, it's not really important for that person to have his salivary glands invested in producing saliva to help him digest the part of the picnic sandwich he may have been eating prior to his encounter with the bear. So the receptors are what determine the response. Let's look at the parasympathetic nervous system. Again, we see the presynaptic neuron releasing acetylcholine, and the postsynaptic neuron is going to release acetylcholine. How do the receptors on these same organs respond? Well, the parasympathetic nervous system is sometimes referred to as food, mood, feed, breed, rest, digest, in which case the receptors in the salivary glands are going to increase their activity. However, in food, mood, feed, breed, rest, and digest, the receptors on the heart will respond differently than the receptors in the salivary glands. Therefore, the heart can slow down its rate of contraction as well as the force of contraction. There are many different types of neurotransmitters that the body uses. We're going to take a look at some of these, and you'll get the idea that neurotransmitters are found in other areas and have different actions. We already know about acetylcholine stimulating the contraction of skeletal muscles. However, acetylcholine is also used by the brain to help regulate memory. Now be careful where you see things or sentences like this. In most instances, acetylcholine is excitatory. The neurotransmitter is not excitatory, it's how the receptor responds to the neurotransmitter. So please be careful with these sentences. Dopamine is a neurotransmitter that produces pleasurable feelings when it's released and is often referred to as being part of the brain's reward system. Dopamine does have multiple functions depending on where within the brain it is released. There's also another neurotransmitter used in the brain called gamma-aminobutyric acid, and glutamate is a very common neurotransmitter in the brain that often causes receptors to act in a manner which is depolarizing. Glycine is a neurotransmitter mostly used by neurons in the spinal cord, and the receptors probably always respond in a way that is not going to spread an action potential from one neuron to another. Therefore, the receptors respond in an inhibitory fashion. With norepinephrine, we know that it acts as a neurotransmitter. Norepinephrine is also a hormone. When you get to the endocrine system, you will learn that the nervous system and endocrine system are intertwined. In the peripheral nervous system, we've already seen that norepinephrine is part of that sympathetic nervous system fight or flight response. It's also used in the brain as part of a neurotransmitter that helps to regulate normal processes within the brain. We've already seen examples of how the receptors on different types of organs can either respond to norepinephrine in an excitatory fashion or an inhibitory fashion. Serotonin is a neurotransmitter that is involved in functions that help to regulate mood, appetite, as well as sensory perception. Serotonin is also released by neurons in the spinal cord and is thought to play a role in having receptors that hyperpolarize in the transmission of pain signals. When we look at the function of the nervous system, we know that when a stimulus is received, such as is in this picture, we have sensory receptors in the skin receiving incoming information that changes the voltage of that receptor, causing a graded potential. If the stimulus is strong enough to reach threshold, an action potential will start to travel down the axon. In this case, we have examples of two different sensory neurons coming from the periphery of the body to the brain. Because we're looking at the sense of temperature with this particular thermoreceptor in the skin, we're going to focus on this particular neuron here, which is sensing information regarding nociception for pain, temperature, and coarse touch. The axon of this neuron will travel to the spinal cord, where it will synapse with another neuron in the posterior horn of the gray matter of the spinal cord. The axon of this neuron is going to ascend because sensory information is ascending and afferent. It will travel all the way up to an area of the brain called the thalamus, where it will again synapse with another neuron, and this neuron will have an axon that will go all the way up to the primary somatosensory cortex, which is where the sensation will be perceived. So again, looking at this initial stimulus, if the stimulus is strong enough, an action potential is generated which travels down the axon. When the action potential reaches the end of the axon, it will release a neurotransmitter. The neurotransmitter binds to receptors on the target neuron, which then has its own action potential. And then this second neuron is going to synapse again, like we said, with a neuron in the thalamus, which then sends information to the primary somatosensory cortex of the brain, where the perception of the temperature of the water begins. That's sensation. That's afferent, incoming, ascending, going upward information. If you look at the lower portion of this picture, you'll see the hand reaching under the water. This was the previous slide where sensory information travels along the axon to synapse with other neurons to take the information to the somatosensory cortex of the brain, where the information is perceived. Remember that the cortex of any organ is the outer portion of that organ. In bone, we saw cortical bone because it comes from the word cortex, which is compact bone, and then an intermedullary cavity. You'll see an outer cortex in kidneys and an intermedullary portion. In the brain, the cerebral cortex is made up of gray matter that consists of neuron cell bodies, or soma, and dendrites. That's the outer candy-coating shell of the M&M. The intermedullary portion of the brain is the white matter. However, there is some gray matter inside. However, when we talk about the cerebral cortex, I want you to remember that's the outer portion of the brain where the neuron cell bodies and dendrites live. This cerebral cortex, by the way, is only 2 to 4 millimeters thick. It's not very thick. So far, we have sensory, afferent, ascending, going upward, information that goes all the way up to the cerebral cortex. Once the information reaches the cerebral cortex, the information is going to be processed. It's going to be integrated with a person's emotional state as well as memories. From that point, a plan is developed and the motor cortex will send out a command to skeletal muscles. The neuron cell body that lives in the primary motor cortex of the brain has an axon that will descend downward through the spinal cord, forming a synapse with a motor neuron, which will take that motor impulse to skeletal muscles. This is descending efferent information. I'm going to remove my picture so you can see this. In sensory information that comes in, there's often at least 3 neurons that have to take information up to the brain, whereas in motor information, there are only 2 neurons involved. There is an upper motor neuron that resides in the cerebral cortex and a lower motor neuron, which has its cell body in the anterior horn of gray matter of the spinal cord. The lower motor neuron is the one that releases acetylcholine onto the synaptic cleft at the motor end plate of the skeletal muscle fiber so that an action potential can initiate muscular contraction. This takes us to different diseases that can occur in the nervous system. Alzheimer's disease is a progressive degenerative disease of the brain that results in dementia. And what happens is the proteins start to fold irregularly, and that forms what are known as amyloid plaques. And these amyloid plaques and neurofibrillary tangles within the neurons basically tangle and kill the neurons, and the brain starts to shrink. There are many famous people who have suffered from Alzheimer's disease. Parkinson's disease generally affects people who are in their 50s or 60s. In the case of a certain actor, Michael J. Fox, it affected him at a much earlier age, which is very unusual. With Parkinson's disease, there is a degeneration of neurons that produce and release dopamine. These neurons are part of an area of gray matter known as the substantia nigra. As degeneration in the substantia nigra occurs, there are other groups of cell bodies in an area of gray matter inside the brain called the basal nuclei that start to become overactive. And this basically messes up with the person's ability to move correctly. So people with Parkinson's disease have persistent tremors while they're resting. They call that a resting tremor. You might see a shuffling gait where they try to start walking and it's like they're stuck. And they also have very stiff facial expressions. Parkinson's disease is treated with L-DOPA, which is a chemical mimic of dopamine. However, it becomes ineffective very quickly over time, and sometimes deep brain stimulation with electrical stimulation can help alleviate these resting tremors. It's kind of interesting that people who have these resting tremors and problems with their shuffling gait and facial expressions are able to ride bicycles very smoothly. There has also been some research done on Parkinson's with using a gyroscope and putting people in this large gyroscope to help to stimulate the apparatus in the inner ear for balance. And they find that that can also alleviate the symptoms of Parkinson's disease. Huntington's disease is a fatal hereditary disorder, and the drugs that are used to treat it are going to block the effects of dopamine. It usually strikes individuals in prior to middle age or around middle age, where there's a protein that has mutated within the neurons in the brain, causing the tissue to die. And what ends up happening is there's massive degeneration of gray matter called the basal nuclei, as well as degeneration of the cerebral cortex. Huntington's disease produces this slow, wormy chorea, or sometimes wild, jerking, continuous flapping, non-voluntary hyperkinetic motions. The disease is progressive. It usually leads to mental deterioration and is usually fatal within 15 years. There are videos that you can watch. Huntington's disease chorea is a video you can look up. I don't have a link for that in Blackboard, but if you're interested in seeing it, you can watch it on YouTube. Creutzfeldt-Jakob disease is a disease that's caused by abnormal proteins called prions that aren't killed by standard methods for sterilizing surgical equipment. When people got this disease, it was mistakenly thought that it was a variant of the prion or abnormal protein that caused mad cow disease, but that's really not the truth. It's just a very common misunderstanding. What happens is, as these abnormal proteins, these prions, start to build up in the cells, it starts to shrink the brain, and the nervous tissue fills with holes to the point that it looks like a sponge. With Creutzfeldt-Jakob disease, the people who are affected by that lose the ability to think, move properly and suffer from memory loss. And you can see the representation on the picture on the right here of the brain shrinking. This disease is always fatal and usually causes fatality within a year of onset of illness. Excuse me. That ends this slide presentation.